Hey guys, welcome back. All right, so I wanted to break up these videos so that you can, not feel too overwhelmed. And it'll also give you a chance to just go back and look over the part that you actually needed to look over. But I am actually just continuing where I picked up at like literally seconds ago. And so the last thing I said was that we're going to talk about the stages of photosynthesis. So let's get ready to science. All right, so there's two stages of photosynthesis, okay? The two stages of photosynthesis are the LDR, which I've mentioned several times already, or the light dependent reaction. And then there's the Kelvin cycle, which is also known as a light independent reaction. Some people actually call it a dark reaction. I don't prefer that term, but if you do hear that, then you'll know what's going on. All right, so the light reaction, of course, happens. We talked about this already. The light reaction happens in the thylakoids, right? And the Kelvin cycle is going to happen in the stroma with the R. Don't confuse stroma with stoma. Stoma or stomata, that's the nose of the plant. In the light-dependent reaction, these are the things that's going to happen, okay? One, water is going to be split. Into hydrogen and oxygen. The oxygen is going to be released, so this is my byproduct. Byproduct doesn't actually have a D in it, but I just thought that it'd be cute to tell oxygen by. All right, and then the hydrogens are actually going to be stripped of their electrons and used in the Kelvin cycle. I put Kelvin reaction. I meant to type Kelvin cycle. So if you were looking at this like, what's a Kelvin reaction? Then my apologies, it's a Kelvin cycle. All right, so the hydrogen ions are stripped of their electrons, okay? So they're stripped of their electrons. And so what you end up having are hydrogen ions plus their electrons that actually will be moved separately to the stroma. In the process, you're also going to reduce NADP plus to NADPH, nicotinamide adenine diphosphate, is going to undergo reduction, and through the process of photophosphorylation, a molecule of ATP is produced. Now, that's important. Both the reduction of NADP plus and the production of ATP are extremely important because these two molecules also move into the Kelvin cycle. They're actually produced in the stroma. All right, they're produced in the stroma as a result of what happens of the structure of the thylakoid and how these electrons are passed, okay? So the hydrogen ions are actually attached and the electrons are attached to the NADP plus to reduce the NADPH. And that's how they are carried into the Kelvin cycle. NADP plus is what I call a carrier molecule, not a keeper molecule. It will pick up hydrogen ions and electrons from one destination and take them to the other. Now, what you also need to remember about ATP is that it is recyclable, right? ATP breaks down to ADP and inorganic phosphate. So the ADP and inorganic phosphate is what's used to generate the ATP. The NADP plus is what you're going to end up with here, and it's going to be oxidized in the Kelvin cycle, but then it is reduced in the light dependent reaction, okay? So NADP plus is going to be oxidized in the Kelvin cycle, and it's going to be reduced in the light dependent reaction. Now, look off the board over here. And so I'm going to come back to the Kelvin cycle, don't worry. So if you look what you have this chloroplast, remember that double membrane enclosed organelle, and so water is going to be split. So water is going to come in, it's going to be split into hydrogen and oxygen. Technically, it's split into hydrogen ions, electrons, and oxygen. The oxygen is going to be released. The energy, of course, is absorbed from the sun, the photons, and they are what's actually triggering all of this stuff to be happening. This is happening in the thylakoids. NADP plus and ADP and inorganic phosphate are going to, ADP and inorganic phosphate are going to undergo photophosphorylation, and NADP plus is going to be reduced, okay? So they are changed into another molecule. ADP becomes ATP and NADP plus becomes NADPH. All of that is happening in the light dependent reaction. Now, the second part is the Kelvin cycle, okay? In the Kelvin cycle, carbon dioxide is actually going to enter into the chloroplast, where it is going to undergo carbon fixation. Remember, we talked about that earlier. It's going to undergo carbon fixation. And during carbon fixation, this carbon dioxide will actually bond with the hydrogens that were carried over by NADPH. When they bond with the hydrogens, they make simple sugars. This is a structural formula, not the actual formula of the sugar that is made. The sugar that is made initially with the Kelvin cycle is G3P, or glyceraldehyde 3-phosphate. Okay, glyceraldehyde 3-phosphate. That's a 3-carbon sugar. All right, and remember, glucose is a 6-carbon sugar, so it takes 2 glyceraldehyde 3-phosphates to make 1 glucose molecule. Now, once carbon has undergone fixation and the sugar is created, how did that happen? What's the energy? What energy was used for this Kelvin cycle? We know that the energy for the light-dependent reaction came from sunlight, right? But where did the energy for the Kelvin cycle come from? Well, the energy from the Kelvin cycle comes from the light-dependent reaction, because that ATP molecule, well, those ATP molecules, it takes more than one, guys, okay? So again, this is a simplification, oversimplification, but just so you'll know where the energy is coming from. So the energy that is generated during the light-dependent reaction, during LDR, is actually going to drive the Kelvin cycle. That's why it's called the light-independent reaction, because this reaction only happens if there's photons present. But this reaction, once it happens, creates ATP, which is an energy shuttle molecule. And remember, ATP can undergo hydrolysis and phosphorylate an intermediate to transfer that energy that was stored within those chemical bonds of that ATP molecule. All right, so that's an overview, believe it or not, of what's happening in photosynthesis. All right, now, let's actually go a little bit more in depth. No, she said the D word. Yes, depth. You know how much I love depth. All right, so let's talk about the light-dependent reaction, okay? So if you look at the board, I'm erasing, so just pay attention to the board and not me. If you look at the board, it tells you that light reactions convert solar energy to chemical energy of ATP and NADPH, okay? So light energy is going to be converted to the chemical energy found in ATP and NADPH. So at the end of the light-dependent reaction, at the end of LDR, the energy can be found in two molecules, ATP and NADPH. That's where you're going to find your energy at the end of the light-dependent reaction. Now, let's talk about sunlight. We know that energy enters into the ecosystem in the form of sunlight and exits in the form of heat, right? Energy transfer is one directional, okay? It is not recycled like other nutrients in the biogeochemical cycles like oxygen or phosphorus or nitrogen. It's not recyclable. Energy enters in the form of light and exits in the form of heat. Because remember, with every energy transformation or transfer, there's an increase in entropy or disorder of the universe, and that is due to that loss of heat into the universe. Now, what is light? Okay, what is light in the first place? Light's a form of electromagnetic energy, and electromagnetic energy travels in rhythmic patterns called waves. So it can travel fast, or it can make big waves, or it can make smaller waves. All right. So these smaller wavelengths. So the wavelength is the one crest to the next one, right? Is it called the crest or the rise? I forget. All right. So this is the length of the wavelength from here to here. Wavelengths. All right. Now, the shorter the wavelengths, the higher the energy. Okay? So the shorter the wavelengths, the higher the energy. So my longer wavelengths are going to have lower energy. I like to think of it as like an EKG, like when you test your heart rate, like when your heart's beating really, really fast, so it's like a lot of energy, your waves are going, like the heart waves are going like boom, 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 boom. But then if it's beating slow, where it has little energy, it's like, you know. So that's what I like to compare that to. All right. The electromagnetic spectrum is actually the entire range of electromagnetic energy, but there's only a small range within that we can actually see. And that small range we can see is called visible light. And so that range goes from about 380 nanometers to about 750 nanometers. And so on the lower end, on the shorter wavelength end, we have like our purple and blues, right? And on the longer wavelength end of visible light, this is where we're going to have our reds and oranges. Oh, I spelled orange all wrong. Y'all didn't see that? All right, so this is where we have our reds and oranges. So again, the smaller the wavelength, the shorter the wavelength, the higher the energy. The longer the wavelength, the lower the energy. All right, so what we actually see though, like when you look at something and you actually see the color, what you're seeing is what's not being absorbed, okay? What you actually see is what's not being absorbed. There are these things called pigments. Pigments are substances that are able to absorb different wavelengths of light. And so when I look at a plant and I see green, it's because the wavelength of light that represents what we call green is being reflected or transmitted through that organelle. All right, it is not being absorbed. So chlorophyll absorbs other wavelengths of light, but not green. All right, now you can measure this with what's called a spectrophotometer, and the way a spectrophotometer works is you have a photoelectric tube and a galvanometer, which galvanometer, I'm screwed that up, you know what I'm talking about, that actually measures the absorption. Well, actually it measures the transmission of light, okay? So you can use a refracting prism, and then you can pass a particular color through a solution, and whatever is being read by the photospectrometer is what's not being used. All right, so when you shine green light through a chlorophyll solution, you'll notice that you have a high transmittance reading. You have a high transmittance reading because the green light is being transmitted through. However, if you did the same thing with blue, your low transmittance because most of that is going to be absorbed by chlorophyll, okay? So this gives you a quantifiable value to the amount of absorption. You can actually use that to measure the rate of certain processes such as photosynthesis with regards to the color of light that you are exposing your autotroph to. Now, a guy by the name of Engelman actually did an experiment, okay? So when we think about plants, there's more than one type of pigment in plants, okay? You have chlorophyll, you also have carotenoids, but even with chlorophyll, there's two types of chlorophyll. You have the main type of chlorophyll, which is chlorophyll A, and then you also have chlorophyll B, okay? So what he did was he took some algae. Yeah, he took algae, and he actually grew it in culture with an aerobic bacteria. Now, why did he use an aerobic bacteria? What's the byproduct of photosynthesis? Oxygen. Aerobic bacteria require oxygen to survive. So he exposed this algae to different wavelengths of light, and he quantified how much bacteria grew in those areas. And what he found was he had huge clusters of bacteria in the wavelengths that corresponded to the purple light, the blue light, the orange light, the red light, but he had virtually no bacteria that grew in regards to the areas where there was green light, okay? So why does the bacteria growth matter? Because when photosynthesis is happening, oxygen is being released, and the bacteria needed oxygen to survive. So the more bacteria you have in the area, the more oxygen that's in that area, and the presence of oxygen indicated that the algae was doing photosynthesis, okay? So that was a way to support the belief that chlorophylls actually work better in a particular color light, and that the reason plants don't grow well with green light is because that was not being absorbed by the pigment. So this is called an absorption spectrum. All right, so this is a graph that actually plots the pigment's light absorption. This is an absorption spectrum. This is an action spectrum, and the action spectrum is actually showing that something is happening in regards to what's going on with the other thing. So what's the action? What's the purpose? What's going on when this light is being absorbed? So you're telling me light is being absorbed in these wavelengths of light, so what's the purpose? Well, it causes photosynthesis to happen. How can you prove that? There's action. There's growth of the bacteria where the oxygen is located, and of course the oxygen is there because it is a byproduct of photosynthesis. All right, let's talk about chlorophyll A and B. So the main photosynthetic pigment is chlorophyll A. Chlorophyll B is what we call an accessory pigment. The difference between chlorophyll A and chlorophyll B is literally what's attached to this carbon. All right, in the head of the chlorophyll molecule, if it is chlorophyll A, there's a methyl group right there. If it's chlorophyll B, there's a carbon, hydrogen, and oxygen right there on chlorophyll B. Also pay close attention to what's at the center. What element is at the center of that head? That's magnesium, right? Now, here's the thing. Doesn't the structure of this kind of resemble a phospholipid, right? And so, which is actually amazing because it has this hydrocarbon tail, which is kind of like the fatty acid tails, that actually interacts with the hydrophobic regions of proteins inside the thylakoid membranes. And that's important because this is a part of the photosystem that actually conducts the light dependent reaction. So what's happening to chlorophyll, all right? Remember that light energy is electromagnetic radiation, electromagnetic energy. So the energy is being absorbed by the chlorophyll molecule, and when that energy is absorbed, the electrons get excited. They go from a ground state to an excited state, which is unstable. All right, and remember, things tend to move towards a point of stability. So as the electrons fall back down from an excited state to the ground state, they actually fluoresce, okay? There's a fluorescence that you can actually see. So it gives off a little bit of light. You can actually, if you could isolate chlorophyll, you can actually see it fluoresce in an isolated solution. And again, every time there's an energy transfer or transformation, some of it is lost to the environment in the form of heat. All right, so light hits electrons and chlorophyll, electrons get excited, they jump up to a higher state, but then they're gonna fall back down. Now, in the light dependent reaction, when these electrons jump up, they're actually accepted by a primary electron acceptor. All right, so they don't get a chance to fall back down to the actual chlorophyll molecule, but they cannot stay in that excited state either. So they're passed down in steps called an electron transport chain to control the release of free energy as it drops from an excited state to a ground state. So I keep saying photosystem and reaction center and all this stuff, but what is that? Okay, so first of all, a photosystem is simply a protein, okay? So a thylakoid itself, so a thylakoid membrane. Remember that membranes are phospholipid bilayer, so of course you have to imagine that this is a phospholipid bilayer. Embedded in the thylakoid membrane, you're going to have two photosystems, all right? You have photosystem 2 and you have photosystem 1, all right? Now, they are named for how they were discovered, not for how they react, because photosystem 2 reacts before photosystem 1. There's also going to be an electron transport chain, okay? So, in your electron transport chain, you have PQ, cytochrome, and PC, which are a part of the electron transport chain. You also have another protein called ferredoxin. And you have an enzyme out here called NADP plus reductase. All right, now, another enzyme is embedded in the thylakoid membrane, and that enzyme is called ATP synthase. Okay, the photosystems have what's called a reaction center and a light harvesting complex. So the reaction center is actually going to be found in the center of the photosystems. The reaction center has, I wish I had a green, I don't have a green. There's going to be a special pair of chlorophyll A's in the reaction centers, okay? Special pair of chlorophyll A's in the reaction center. These chlorophyll A's have a name. Uh-oh, went the wrong way. I'm trying to go yet. All right, so this one found in photosystem 2 is called P680. And that's because it works best when it absorbs 680 nanometers of light. This one is called P700. Guess why? All right, it works best when it absorbs 700 nanometers of light. All right, now in the reaction center, there's also what's called a primary electron acceptor, okay? So both of these have a primary electron acceptor. I want to draw your attention to this one, the one found in photosystem 2. This is called P680 plus. and it is the strongest oxidizing agent known to man. It's the strongest oxidizing agent we know. All right, so what does an oxidizing agent do? An oxidizing agent is going to be something that allows something else to lose its electrons. So an oxidizing agent is there to accept electrons so that whatever is giving them away can be oxidized. The oxidizing agent itself is going to be reduced, okay? So the thing that is reduced is called an oxidizing agent. The thing that is oxidized is called a reducing agent. All right, so in the photosystem, so we have, this is the reaction center, okay? So this part right here is called the reaction center. Remember, the whole thing is a photosystem, right? So this center part is called the reaction center, and this part right here is called the light harvesting complex. So guess what it's gonna do? Harvest light. And it does that by having these pigment molecules, right? So these pigment molecules are located in the light harvesting complex. I forgot to mention, I know it was on the PowerPoint, I forgot to mention, other than chlorophyll A and chlorophyll B, there's also other pigments called carotenoids. I may have mentioned those, but the purpose of carotenoids is actually to, they function in photoprotection. Carotenoids actually absorb additional wavelengths of light that could damage the chlorophyll. So they function in photoprotection. So keep that in mind too, when you think about the pigments that are found in chloroplasts. Now, so see if you can follow along with me, all right? So what's going to happen? Light energy in the form of photons captured by the pigment molecule passed from pigment molecule to pigment molecule until it reaches P680. Remember, these guys right here are a special pair of chlorophyll A, all right? Now, what do we say happens when chlorophyll gets light energy? The electrons get excited. So the electrons that were here get the energy, they get excited, and they jump up to a excited state where the primary electron acceptor is all too happy to accept them. Now remember, an excited state is unstable, all right? So when the electrons get to the primary electron acceptor, they still need to fall down to a ground state, but that fall is controlled by passing the electrons from protein to protein. This is called an electron transport chain, okay? This is called an electron transport chain. Finally, the electrons are passed to photosystem 1, where they sit until the pigment molecules that are located in photosystem 1, because they both have pigment molecules in the light harvesting complex, when they absorb light energy, they do the same thing, pass it to the special chlorophyll A, and these electrons get excited yet again, jump up to another primary electron acceptor, okay? Jump up to a second primary electron acceptor. What is this one? P700 plus, right? Because if this one is P680 plus, this one is P700 plus. Now, this primary electron acceptor has to pass the electrons through yet another electron transport chain. This time, the electron transport chain is going to include ferredoxin, and ferredoxin is going to pass the electrons to an enzyme called NADP plus reductase, okay? So this is where your enzymes, your electrons that were a part of P680 end up. Now, here's the problem. When I did that, when P680 underwent the reduction, it lost electrons. It needs the electrons back. How do we solve that problem? Enter water. Enter water. Enzymes are used to split water the oxygen was not the target of this reaction, so the oxygen is released, right? So we get half of the O2, because remember, atmospheric oxygen is O2. So the oxygen is released, which leaves us with these hydrogens. What do hydrogens have? One proton and one electron. We were missing how many electrons? 2. So hydrogen is stripped of its electrons. The electrons that were in the hydrogen replace the electrons that got excited from P680. So when you take hydrogen's electrons, you're left with what's called a proton, because it's hydrogen with just a proton. So you end up with a buildup of protons inside of the thylakoid space. That's the space. So if you look at the thylakoid, you have the membrane, and then in here, you have space, okay? So you end up with a high concentration of the, protons on the inside, and you also have some protons being pumped back in from the outside into this space. That creates what's called a proton gradient, all right? And how do things like to naturally move, right? So if I have a high concentration of protons here, why did I put a P? Concentration of protons here, and a low concentration of protons here, I created a concentration gradient where things naturally want to go from an area of high concentration to low. Remember, we talked about transport, but this is an ion, so it cannot simply cross the membrane, so it has to go through a protein channel. That particular protein channel is called ATP synthase. This is an enzyme. Remember, enzymes are proteins. So the hydrogen ions, because of the gradient that is being built up as these electrons are being passed, because you have to replace them by stripping the electrons and hydrogen, so that builds up my protons on the inside. They're going to move from high to low. They go through what's called ATP synthase. ATP synthase is an enzyme that catalyzes ADP, an inorganic phosphate, transformation to ATP. So this is where you get your ATP molecule from, okay? So at the end of the light reaction, what you're going to have is these protons. Oh, I forgot to mention this part. The protons, here's the cool part. The protons that end up out here, right? Remember, they are missing their electrons, right? So the electron transport chain has passed these electrons to where? NADP plus reductase. So guess what these protons do? They're moved to this enzyme. This enzyme's job is to join NADP plus with the hydrogen ions and the electrons, right? So what you end up with is NADPH plus a hydrogen ion. So at the end of the light-dependent reaction, at the end of LDR, the energy is going to be found here and here. ATP and NADP plus. Now, here's the thing. You need both of those for the second reaction, the Kelvin cycle. The Kelvin cycle happens in the stroma. Look at where they are. They're outside of the thylakoid already, right? So ATP is actually generated in the stroma. NADP plus is actually generated in the stroma. So they're already in the location they need to be for the second reaction, all right? So I know that's a lot, but I love it. So photosystems 1 and 2, of course, everything you need to know is here and here. Let's look at it in a better drawing, right? This is your photosystem. In the middle, you have your reaction center. Your reaction center is going to have your special pair of chlorophyll A's and your primary electron acceptor. Outside, you have the light harvesting complex with your pigment molecules, okay? So This flow that I did on the board is called the linear electron flow. This is the way that most plants do this. Now, here's the killing part. Here's the funny thing. What are the two things I made in the linear, right? So this is the linear flow. In the linear, I produce ATP and NADPH as my energy molecules, and then I produce oxygen as a byproduct, okay? There's also a cyclic flow, and in the cyclic flow, the only thing I'm going to make is ATP. The cyclic flow does not make NADPH, nor does it release oxygen as a byproduct. But okay, so let's go back through in pictures what I drew up here. All right, so look over here at the board. Photosystem 2, light harvesting complex on the outside, reaction center in the middle. See your P680 is your special chlorophyll A, primary electron acceptor, P680 plus. So light is passed from pigment molecule to P680. Electron gets excited, hopped up to an excited state where it is accepted by P680 plus, your primary electron acceptor. Now, this is going to show you the immediate putback. I followed the electrons all the way through, same thing, but this is going to show you how water is going to be split to replace the electrons that got excited from P680. Oxygen is released as a byproduct, and you have created these hydrogen ions, okay? The primary electron acceptor is going past the electrons, because remember they're at an excited state, they have to fall down to stability at the ground state. So to control the release of free energy, everything that's done in steps is done to control the release of free energy. It's going to be passed through an electron transport chain, and ATP, of course, is going to be created as a result of the proton gradient that is created. I'm sorry, I lost my train of thought. All right, so ATP is going to be made as a result of the proton gradient. The electrons end up in P700, which is in photosystem 1. All right, that is another in the other reaction center. Light is going to excite the electrons again, and they hop up to P700 plus, which is the second primary electron acceptor. It's the primary electron acceptor for photosystem 1. From there, they enter another electron transport chain where they end up passing from ferredoxin to NADP plus reductase. Now, the hydrogen ions that were a part of water will actually be catalyzed in a reaction, will be a part of the reaction that is catalyzed by NADP plus to join NADP plus with those hydrogen ions and those electrons to make NADPH. All right, so steps written out here. Just another way of looking at it. Let's talk about the cyclic flow. You already pretty much know it. The only difference is it does not involve photosystem 2, okay? The light excites the electrons in P700. They get excited, jump up to the primary electron acceptor, which passes them to ferredoxin, but ferredoxin passes them to a cytochrome complex, where they go from the cytochrome complex to PC back to the P700, okay? So that's a cyclic flow. This happens over and over and over again, and the only thing that is produced in the cyclic flow is ATP. Now, some older plants don't have photosystem 2, they only have photosystem 1. So they use that to produce ATP. And cyclic flow can also happen, it can occur in plants that have both, and it may be to protect the cells from light-induced damage, okay? To pull in that 700 nanometer light waves and actually use it for something. All right, so here's an overview of what I attempted up here. So what you have, entire thing, photosystem 2, you have your electron transport chain, photosystem 1, you have ferredoxin, NADP plus reductase, and you have your ATP synthase right here. And all of that is going to go on to the Kelvin cycle. And the Kelvin cycle is what we will actually do in a synchronous meeting on Wednesday. So see you guys. Toodles.